Hello, my name is Ross Cummings and I am UK and European patent attorney at Gill James and Every, a leading firm of patent and trademark attorneys in the UK and Munich. This is part 3 of this expert talk. In parts 1 and 2, I've talked about patented gene therapies in general terms, but I thought it would be interesting to look at a couple of specific cases to provide some context before we finish. And where better to look than the only 2 gene therapies to have achieved regulatory approval in Europe and the US, Luxturner and Zorgantomate. The earliest patents for these 2 therapies provide a nice contrast, showing instances of narrow and broad claims respectively. The first of these was Luxturner, which is based on work that was filed in a patent application in 2002, which is co-owned by 3 universities, Pennsylvania, Florida, and Cornell. Luxturner is used to treat adults and children with loss of vision due to inherited retinal dystrophy, a rare genetic disorder of the retina, which is the light-sensitive membrane at the back of the eye. Luxturner can only be used when the patient still has enough functioning cells left in the retina, and the disease is caused by mutations in the RPE65 gene. This gene is responsible for the production of an enzyme called all-trans-retinal isomerase, which is necessary for the normal function of retinal cells. Luxturner consists of adeno-associated virus 2, carrying a normal copy of the RPE65 gene. Now, the 2002 patent application was filed in the US, Europe, and Canada, and when it was examined in Europe, it had very broad claims. Paraphrasing, the application claimed a composition suitable for treating any ocular disorder identified by the defect or absence of any normal gene, the composition comprising any recombinant virus carrying the normal copy of the gene under the control of any ocular promoter, formulated for subretinal injection. A dependent claim specified the AAV and RPE65 combination. Other genes are specified in other dependent claims as well. Claim 1 was not novel over known vectors carrying different genes, but the RPE65 in combination with an adeno-associated virus was novel at that time. To support the invention, the application contained data that demonstrated construction of adeno-associated virus carrying canine RPE65 with chicken beta actin promoter CMV enhancer. And then they showed strong expression of the RPE65 following in vitro infection of canine retinal pigment epithelial cells. And then they showed detailed and extensive canine in vivo studies with mutant RPE65 showing long-lived expression of the transgene in the right parts of the eye and no detectable extraocular expression. And finally, they had canine functional visual testing showing restoration of vision following subretinal injection at certain doses, but with no effect with IV injection. So this is very complete and excellent data, but only for a specific vector and showing effect at specific dosages via a specific delivery route. Sufficiency and plausibility were not raised by the Patent Office, partly because plausibility is a more recent product of case law, and also because the data in the application easily met this requirement anyway. The EPO examiner objected that the broad claim was not novel and that the AAV and RPE65 combination was obvious, as there was known proof of principle for ocular gene therapy using recombinant AAV, and adenovirus had been used with RPE65 in canine studies. Now, it's not clear why, but the European application was abandoned shortly after the start of examination. In the US, claims were granted to the specific construct, disease, and dosage shown to work in the canine studies. It is likely that similar claims could have been granted in Europe. The granted US claims nicely illustrate limitations you often have to make around the experimental data when working in the gene therapy space. Now, the take-home message from this is that your patent may be limited to the specific vector and dosage that you tested. So if you find that after filing a patent application, you have made tweaks to your vector or dosage that you intend to take forward to clinical trials, you must ensure that you file a follow-on patent application to capture the tweaked vector, no matter how subtle the change. So that that may lead to a very narrow patent, but this narrow patent to the final candidate could be the difference between no patent protection and very relevant long-lived protection for the marketed product. Ultimately, inventive step will depend on whether you show that specific product to be efficacious. You may not know this until after the clinical trial, so you need to have the applications on file before you do those trials. I'll just briefly consider Zorgantomate, which is used to treat spinal muscular atrophy, a serious condition of the nerves that causes muscle wasting and weakness, mainly in babies and small children. This is caused by abnormalities in the human survival motor neuron gene, SMN1, or multiple copies of SMN2 in a variant of the disease. Zorgantomate uses a vector that is based on serotype 9 AAV, but with an important difference. It uses a self-complementary AAV9 that is effective at crossing the blood-brain barrier and expressing genes in the spinal cord. Self-complementary AAVs represent a very clever solution to the problem that AAVs have single-stranded DNA genomes, but mammalian cells have double-stranded DNA as the basis for expression. In self-complementary AAVs, a single-stranded genome contains a so-called inverted terminal repeat genome that can fold on itself to form a double-stranded DNA upon delivery. This skips out the otherwise necessary initial DNA synthesis step or complementary strand recruitment step for another AAV before gene expression can commence. This means you don't actually need as many vector particles to be delivered to the cell, which can dramatically increase efficacy and address dosage problems. The vector used in Zorgantomate was the subject of a patent application filed by Genethon and the French National Centre for Scientific Research in 2008, which is granted in Europe and Japan and the US. It was actually opposed in Europe and is under appeal. This patent granted with broad claims to an AAV vector comprising a gene of interest and either being a double-stranded DNA self-complementary AAV vector or a pseudo-typed self-complementary AAV vector having a capsid from AAV9. So the claims cover the use of any transgene with this vector in the context of neuromuscular disease. The inventors discovered that these self-complementary AAV9 based vectors were particularly good at delivering genes to motor neuron cells or glial cells. This activity was supported by murine and feline in vivo models showing expression of a reporter gene in spinal cord tissue sections. So the patent application clearly demonstrated that it was plausible that these vectors could carry a transgene to the spinal cord and drive expression. In summary, while there appear to be significant hurdles to overcome to obtain patents in gene therapy in Europe, there are many opportunities for effective protection. You need to ensure that your scientists and regulatory teams are well aware of the patent challenges and keep you in the loop with developments. Otherwise, valuable IP may be missed. Regulatory exclusivity is also very important in this field, like data exclusivity, market exclusivity, and orphan drug exclusivity. These rights are going to be helpful at the point of approval, but they do not provide protection early on in fundraising and development, and do not prevent competing companies from seeking their own approval, rather than relying on your data. And due to ethical hurdles in getting approval for clinical trials, particularly in orphan designations, it's crucial to be first to the clinic. Therefore, an effective patent strategy is always going to be important. Patent extensions through supplementary protection certificates, which are not discussed here, are also available, but only for product claims. And thank you very much for listening. If you have any questions or concerns, then please get in contact. On the details are here. And have a good day. Goodbye.